Right, are you guys ready for the last lecture for microbiology for this semester? This is our last one. It is the cardiovascular and the lymphatic system diseases. And so next time we quote unquote meet again will be for the final. Okay, and so I've talked to you guys about the final before. 25% is going to be the disease unit, 75% is going to be everything else from the semester. I am working on a review sheet. It'll probably be posted by the time you guys watch this video, but there will be a review sheet for the disease unit on D2L, and the others you will use your previous review sheets, to be studying. Oh, goodness. So begin studying immediately if you haven't already. You know there's a lot of material here, so be very diligent in putting in the time necessary to study. Okay, so chapter 21, cardiovascular system and the lymphatic system. Let me back up one. So I always tell the A&P students that the cardiovascular system includes two major structures. The cardio part is the heart, and then the vascular part is going to be the blood vessels. And then earlier, when we were talking about the immune system, we very briefly covered what the lymphatic system is, so you should already kind of have an idea what that is there. So infections in the cardiovascular system, or the lymphatic system can readily spread throughout the body, right? Because those two systems are made up of vessels that go throughout the body and can cause spread very easily if there is infections present there. In the cardiovascular system and the lymphatic system, if there is an infection, that's called a systemic infection because it's probably going to spread throughout the body. And a lot of times systemic infections began as a localized infection, okay, or an infection in one particular part of the body. They entered the bloodstream or the lymphatic system and spread from there. Okay, so just really quick review on the heart. The heart has 4 chambers. Hopefully my anatomy physiology graduates or those that have had it this semester can remember these. There's the right atrium, the left atrium, and those two make up the upper 2 chambers, and then the lower chambers are the ventricles. And so the atria receive blood, the ventricles pump blood out either to the body or to the lungs in the case of the right ventricle. And then the heart is made up of several layers. There's the inner lining called the endocardium. The muscle is called the myocardium, and then there's the pericardium on the outside. Now here's a couple of terms. These will be on your review sheet. Sepsis, okay? You've probably heard that term before. If someone develops sepsis, that's kind of a scary situation. Sepsis is not an infection, and in fact, it's actually an immune response to a toxin or a pathogen, okay? So, I'm so sorry. Oh, goodness. Sepsis, if it continues, if that immune response continues, it can progress into septic shock. Okay, so septic shock is basically a complication of sepsis. And sepsis, when it leads to septic shock, you can experience low blood pressure. And because of that, oxygen and nutrients can be cut off. And when that happens, there could be organ failure and necrosis. And if you want to jot down, necrosis is basically death of tissues. They're basically starving for oxygen and nutrients. Okay. And another thing that happens during septic shock is disseminated intravascular coagulation. So intra means inside of. So inside of the vascular system or inside of the blood vessels, there is coagulation or blood clotting that occurs, and that is again attributed to an immune response, and that can block blood flow and just add to that organ failure and necrosis. Again, because you have poor blood pressure, and then if you block those vessels on top of that, that makes things even more difficult. So bacteria or their toxins can cause, or a lot of times can cause sepsis. Typically, the most common are going to be E. coli, staph, and strep. But there's about a third of all cases where the actual pathogen isn't identified specifically. So here are some sources of sepsis. Perhaps a lung infection like pneumonia, a UTI, some sort of gut infection, and that crossed over from the gut, or a skin infection. And so again, remember, a lot of these systemic infections or sepsis starts out in a localized area, and then the pathogens or the toxins spread into the bloodstream or the lymphatic system. And so kidney and lung infections have the highest risk of spreading to the blood when you compare to other localized infections. Sepsis is a medical emergency because it can progress into shock, and shock is very important to take care of that, because there's anywhere from about a 1/4 to a 50% mortality rate with that. Sepsis is very costly to treat because typically the person is going to be in intensive care, be intubated, and have some severe care and a long road ahead of them to recover. And so it is a major threat. Individuals with an increased risk for developing sepsis might be someone with a chronic health issue like cancer, diabetes, AIDS, or people that have had surgery recently. So typically, if a person has sepsis, they'll have at least 2 or more of these signs. And one of them is either a fever above 101 or a low fever, hypothermic, at below 97 degrees. A rapid heart rate, rapid respiration rate, and increased white blood cell count if they did a blood draw. Some other signs and symptoms that may be included in the diagnosis: diarrhea, vomiting, your skin may be pale, tiredness, confusion, and then lymphangitis, which is this here, those red streaks, and that is basically inflammation of a lymphatic vessel. Okay, so if the sepsis involves the lymphatic system, you may have those red streaks there, and this is the path of a lymphatic vessel through the arm. Diagnosis, looking at blood draws, high white blood cell count, high lactate levels, high reactive or C-reactive proteins, low blood oxygen. And then they could also culture the blood and see if there's anything growing in the blood. We used to believe that the blood was azenic, which means did not have anything growing in it, like the cerebral spinal fluid. But there can be very few, very specific microbes that live in the blood. Not very many though. And if we would have been able to go to the PMC hospital lab, one of the machines that we would have gotten to see is their machine that does blood cultures. And if they're testing someone for sepsis and doing a blood culture, they have these, I don't know how many milliliters they contain, but these large vials. They will draw a large volume of blood, and then they will stick those vials of blood into this incubation unit, essentially, and grow them for 5 days. And there's an alarm and everything that will go off if the machine detects microbial growth. So that's one of the things that they do to confirm if a person is septic or not. And so treatment would include anti-inflammatory medications, getting the body temperature back to where it should be, getting blood pressure and oxygen levels where they should be, and then if appropriate, using antimicrobials. Now remember, gram-negatives have endotoxins, so if it's caused by E. coli or some other gram-negative, you may not want to give a high dose of antibiotic because that will even worsen the shock, the immune response. Okay, so then we talked about life support also as well. And I'll let you guys look over this. And then we will move into the viruses. And I've run out of room kind of up here, so I'm going to write over here to the side. And these viruses right here that are listed here, dengue fever, yellow fever, chikungunya, and Zika, and also West Nile virus. And then we have Eastern equine encephalitis, we have Western equine encephalitis. These are very rare, but they can be transmitted between mosquitoes and humans and horses. And there was an outbreak last summer of Eastern equine encephalitis in humans in Ohio. So anyway, I was just mentioning that because there was a slight outbreak last year. But these are all considered arboviruses. And arbo stands for arthropod borne, which is the BO viruses. So most of these are transmitted by mosquitoes, which are a type of insects, are a type of arthropod. So mosquitoes, as in our ticks, are important biological vectors. If you remember that term, there's mechanical and biological vectors. The mechanicals, like a fly or a cockroach carrying one thing on its body to a pathogen from the bathroom to the food prep areas or whatever. But the biological vector, remember, is a part of the pathogen's life cycle. Okay, so these would be biological vectors because the mosquito in these cases are going to be involved in these viral life cycles. Okay, so Aedes aegypti is a mosquito that can transmit arboviruses, and it's found in areas in the South in the United States. It thrives in urban areas. It prefers human blood meals over other animals. It can bite at any time of day instead of at dusk and dawn and night and so forth, like some of the other mosquitoes. And one arbovirus caused disease is the Zika virus. And Zika was first identified in Uganda and Tanzania back in 1952. And in the United States, the first cases began in 2016 in Florida. And so this disease is transmitted between mosquitoes and humans. And humans can spread it to other humans through body fluids, or it can cross the placenta, and this is where it does the most damage, is when it crosses the placenta and infects the fetus of a pregnant woman. Some signs and symptoms, a lot of times they're going to be asymptomatic, but symptomatic patients would have fever, joint pain, this rash, and red eyes. And the major complication is going to be microcephaly, and that is where the baby's born, the head circumference is significantly smaller than the normal circumference of an infant, and so it causes some issues with fetal development, can even cause miscarriage. And then Guillain-Barré syndrome is another complication that can develop because of Zika. And so it's a very, very scary disease for pregnant women, because you don't want your baby to have birth defects. And so it's recommended that a woman should wait for 8 weeks after infection to conceive, and a man should wait for 6 months to father a child after infection. And why there's a difference in timeline there, I'm not really sure. May have to do with how the virus is harbored in males versus females, but I really don't know why that's different. But we do not have the Aedes aegypti mosquitoes here in Colorado, and so unless you travel to an endemic area, you don't have to worry about Zika virus here. Snapshot incubation period is 3 to 10 days. It's also found in Asia, Pacific Islands, South and Central America, Africa, and India. Can be spread through sexual contact. Here's where it says the 8 weeks and 6 months. Okay. Next, we'll get into some of the viral hemorrhagic fevers. Okay, so these were West Nile, Zika, and so forth are the yellow fever, or the arboviruses. These are a class of viruses called hemorrhagic fevers. And hemorrhagic, as you probably can guess, if someone is hemorrhaging, they're bleeding. But a hemorrhagic fever or hemorrhagic pathogen is going to cause, obviously, bleeding, because the pathogen causes an interference in blood clotting, or the clotting coagulation cascade. Hemorrhagic fevers, there's a few of them. We're just going to look at one. Include high fever, chills, diarrhea, vomiting, headaches, and then bleeding from different orifices and internal organs, because the ability to clot has been disrupted. Depending on the virus, can be transmitted by mosquitoes, rodents, bats, or other animals. And the one, we're going to talk about Ebola virus, but hantavirus is one, and there's Lassa fever, and there's also Marburg fever. And if you've studied mortality rates surrounding this COVID thing and how COVID compares with mortality rates with others, you may have come across Marburg fever, and that one has an 80% mortality rate. So 80% of people that contract that will not survive. So that's one of the highest, maybe even the highest disease with as far as mortality rates. Actually, no, it's not. Rabies is. So hantavirus is something that is potentially around in Southeast Colorado, probably more Southwest Colorado, but it could be a possibility. That is where, there's a specific type of mouse, and it just left me, that carries the virus and excretes the virus in its excrement. And then typically when you would get hantavirus is when you're cleaning a garage or cleaning a very dusty area that's been littered with mouse excrement. That dust gets the virus gets aerosolized, and you breathe it in, and it causes a hemorrhagic condition. But it's very rare. Okay, so the hemorrhagic fever we're looking at is going to be Ebola, and it and Marburg virus are caused by a filoviridae virus called Ebola virus, and it's a single-stranded RNA virus. In case you were wondering about that. Remember, viruses have very unique genetic makeup as far as DNA or RNA, which is very unique, and it could be single-stranded or double-stranded DNA or RNA. Ebola can be transmitted by fruit bats, other primates. Those are suspected to be their natural reservoir where the virus normally hangs out. And so a person could get it if they come in contact with the animal's excrement, if they eat those animals, or it can be spread person to person. And there is a video on this that you guys will watch about Ebola. And it'll give you a little bit more of an inclination of how it is spread. But Ebola has two kind of manifestations or two progressive steps in the disease progression. The first one is the dry phase, okay, where there's a lot of weakness, a fever, sore throat, headache, muscle pain type of thing. And then that will then progress into the wet phase, which is the wet refers to blood. So when they cough, they may get blood coughing up, bloody diarrhea, blood in the vomit, may have a rash, and then eventually there will be severe internal and external bleeding. The person will then, because of the bleeding, will go into hypovolemic shock, which is a low blood volume, and organs will start to fail. Inside of the host, the virus likes to attack the macrophages and the dendritic cells of the innate immune system. This is going to cause a huge inflammatory response, cause a lot of inflammation throughout the body. And this virus can attack other cells as well and impact the kidney, the liver, the lungs. And the mortality rate is about 50%. So death occurs in about 50% or half of all infected individuals. Treatment is going to be supportive therapy. There is no antiviral at this point. So keeping the person hydrated, helping with the fever, basically those supportive type things. There is a vaccine that's in the development processes. And the good thing is that if you are a survivor and you're exposed to the same strain, you have immunity toward that strain. And the most recent Ebola outbreak was in 2014 and 15 in West Africa. It was the largest outbreak recorded with 27,000 cases. And again, that approximately 50% mortality rate. You can see the number of cases per area here in Africa. And let's see, incubation period, 2 to 21 days. So that kind of varies a little bit there. Death occurs by shock, that hypovolemic shock most likely. What else did I have here? Oh, diagnosis is difficult. Early diagnosis is difficult because there's a lot of vague type symptoms. The fever, the sore throat, and those types of things can point to other diseases as well. So, but if there's an outbreak, that's probably what they would think at first. Okay, so if you want to draw, hold on. Some other viral diseases of the lymphatic system and also the cardiovascular system is going to be mononucleosis. And that's going to be caused by the Epstein-Barr virus, which is a herpes virus in that herpes family, herpes viridae. And then there's HIV, and we've talked a little bit about that one before, a human immunodeficiency virus. It attacks helper T cells. And so it then weakens the immune system, and usually the cause of death is going to be some opportunistic type infection. HIV is also what we call a retrovirus, because it's an RNA virus that can make DNA because of its enzyme reverse transcriptase. Okay, so these are bacterial infections, if you want to draw a line through there. And we're starting out with systemic bacterial infections, again, getting through the lymphatic or circulatory systems. Again, could come from a localized infection, an IV or catheter, or arthropod bites. And the term bacteremia is the mere presence of bacteria in the blood, and that can, if bacteria are there, there can be an inflammatory response that develops, and that can progress to sepsis, which can progress to shock and death. And so we talked about the blood sample being collected in the special machine, and typically when we do like a throat culture or a urine culture, those are done in 24 to 48 hours, and these are done for 5 days just to make sure. The first bacterial disease, the specific bacterial disease, is going to be the plague or Black Death, which is caused by a gram-negative microbe called Yersinia pestis. And it likes to live in rodents and bats and such, and it can be transmitted between the rodent or the bat with fleas. Okay, so it can go from the rodent to the human through flea bites. And so you've probably heard of the plague before in history classes and so forth. And there were two major incidences of plague back in the Middle Ages, the 1300s and the 1500s, where a significant portion of the European population was wiped out. Like many people died because of this pandemic, these pandemics. There are some sporadic cases that occur worldwide each year. Here's a few cases that occurred in the US. I believe this was 2015, if I remember correctly. You can notice there's a few cases here in Colorado, and Oklahoma Panhandle. And a lot of times the plague comes from prairie dogs and infected fleas associated with prairie dogs. The fleas come and, or your cat or your dog goes out and walks through a prairie dog town, picks up the fleas bite the dog. Why pestis doesn't cause disease in dogs or prairie dogs or other rodents, but it comes back, you pet the dog, the flea jumps off and bites you, now you have the plague. So transmission is through a bite from a flea or the rodent itself. Okay, and we'll talk about human-to-human contact here in just a second. The other thing that I wanted to mention about the fleas is that the Yersinia pestis bacteria causes kind of like a pseudo membrane across the esophagus or the feeding tube in the throat of the flea. And so the flea is really hungry and it's starving, and it bites the host, it bites the rat, and the rat is has blood and it has meal for the flea, but the flea can't swallow its meal, so it gets agitated, and so it'll bite the rat several times, and then it gets frustrated, and it might bite to a human and bite it several times, and it can't get a meal, and then it'll get frustrated and jump to the next person, and each time it's biting, it's transmitting the bacteria, but it's not able to get a meal. So basically the bacteria cause the flea to hyper spread more of the bacteria around into different hosts because the flea is starving. So anyway, I thought that was really crazy. But there's 3 forms that the plague comes in: pneumonic, bubonic, and septicemic plague. As you could imagine, the pneumonic plague infects the lungs. Okay, and this one can be transmitted person to person. So if a person with pneumonic plague coughs, if their lungs are infected and they cough on a caregiver, they can transmit the plague, the pneumonic plague, to the next person. Bubonic plague, though, is the most common form, and that would come from the bite. And this person may have gotten pneumonic plague. So bubonic can progress into pneumonic plague. Okay, so they would have gotten it from the rodent. That can progress there, and then it can spread to another human in this way. The bubonic plague infects the lymphatic system, and they call it a bubonic plague because it produces what's called a bubo. And a bubo is basically an enlarged area of the lymphatic tissue. Usually could be in the groin area or other places where there's a lot of lymphatic tissue. It's basically this big hard mass that's the inflamed or enlarged lymphatic tissue. And then septicemic plague is when the bacteria enters the bloodstream. And so either one of the other two types can progress into septicemic plague. And if you've ever wondered why they called it the Black Death, right there is your answer. It's called acral gangrene. Okay, so we talked about gangrene caused by Clostridium perfringens. This is another type of basically necrotic tissue caused by the septicemic form of the plague. And so if a person has bubonic or pneumonic plague, that plague can then spread to the bloodstream if it's untreated and cause fever and chills and diarrhea and so forth. And then the inflammatory responses can progress into that disseminated intravascular coagulation, that blood clotting, low blood pressure cuts off blood flow to the tissues, and extremity tissues are going to be the first to be affected. And they're going to start developing necrosis, or the tissues are going to start dying and turning black. And for this patient to survive, he's going to have an amputation. Okay, but that's why they call it the Black Death, because back during the pandemic plague times, there weren't antibiotics. Those patients a lot of times did get black tissues. So mechanism of disease, bacteria gets in the bloodstream. The neutrophils or macrophages, those phagocytes, consume or phagocytize the bacteria, but the bacteria aren't destroyed by the phagocytes. And so the bacteria can reproduce within those phagocytes, and they're kind of hidden there. It's one of their virulence factors. And then when the phagocytes, the macrophage or the neutrophil dies, it spreads, and it's probably moved from the site of first infection. And so it's transmitted the bacteria to another location. I mentioned amputation. Septicemic plague is fatal if it is not treated. Gentamicin and Cipro, remember that antibiotic that we did the antibiotic product sheet on? Those two are good choices for antibiotics for Yersinia pestis. Okay, another way that bacterial diseases can be spread are through ticks. And ticks and mosquitoes are probably the biggest vectors of disease. And so ticks, this is kind of interesting. I wasn't really aware of this too long ago, but ticks need to stay on a host for the most part 1 to 2 days, or 24 to 48 hours. That's a 48, by the way, to basically regurgitate enough bacteria to cause infection. So basically what a tick does is it climbs on the host and basically latches on with its mouth parts. And just like a mosquito or some of the other external parasites, they don't want to cause discomfort to the host. Because if a person feels discomfort and like, oh, there's this bloodsucker on my arm here, they will immediately remove it. So ticks basically have learned to crawl into hidden places, like maybe on the neck or near the hairline or underneath clothing and things like that, to first of all hide, but also when they feed, they regurgitate or basically spit into your bloodstream or into your tissues. Anticoagulants, so the blood will keep flowing, and kind of an analgesic as well, so you don't feel it. But when they do that, if they are carrying the bacteria, they will regurgitate that bacteria into your system as well, and that's how they get into the body. And so typically they need to have been feeding on a person for at least a day for you to get enough bacteria to cause infection. Now, if you've only had a tick on you for a little bit of time, does that guarantee that you're not going to get a disease? Not necessarily, but for the most part, that does. And the best way to prevent tick infections or infestations or whatever, getting ticks on you, is to wear light colored clothing if you're going to be in the woods or in an area where there are ticks, so you can see them. They're dark in color and you can brush them off, or using basically DEET or other type of repellent. But again, they like to get in the hair in places where you don't normally, and my hair got all crazy, really yucky today. Different places where we won't necessarily notice them. So we're not going to cover all of these, but some common tick transmitted diseases is going to be Lyme disease, Rocky Mountain spotted fever, we'll cover both of those, ehrlichiosis, and anaplasmosis. Lyme disease is caused by Borrelia burgdorferi, and it is a gram-negative spirochete, so it has a spiral shape. Infects about 300,000 people in the US every year. Its reservoir is the white-footed mouse, and it is transmitted by the deer tick, which is a very, very small tick. Typically, you start to see signs and symptoms about a week or two after infection, and 3/4 of patients that have been infected are going to have this bullseye rash that's very typical of Lyme disease, and it's called erythema migrans. And this would be where the tick was attached, and it forms this type of rash here in this example. So this infection spreads fairly slowly, and it goes through the lymphatic system, causes fever and headache and so forth. And left untreated, this can lead to arthritis. It can cause some chronic damage because of the inflammatory response that can lead to arthritis. And that is how they discovered Lyme disease in the 1970s. There's a particular county in Connecticut called Lyme County, where they noticed that there was such a high rate of childhood arthritis. Childhood arthritis is very rare, and they noticed there's a significant portion of the population of children in that particular county that had Lyme disease. And so they got to studying this, and they determined that the bacteria was being transmitted through tick bites and causing the Lyme arthritis to develop in the children. And so that was the first time Lyme disease was ever identified. Some other complications could be irregular heartbeat, inflammation of the muscle layer of the heart, and also neurological problems. Diagnosis would be based on that rash. There's other blood tests that can be done, and doxycycline would be a drug of choice. Rocky Mountain spotted fever is another tick-borne disease caused by rickettsia, rickettsia. It is an intracellular gram-negative bacteria, so it lives inside of the cells and not necessarily on the outside. It occurs in all parts of the US, but the major places where you see it is around the Appalachian Mountains and not the Rocky Mountains, so that's kind of a misnomer there. But it is a tick-borne disease, and the spotted fever gets its name from the rash that has kind of flat, pinkish red spots on the skin that's not itchy. It can start out from the wrists and arms or ankles and move toward the torso. It's going to infect endothelial cells, and if left untreated, it can be fatal within 8 days. And the reason that it's so fatal is it can cause damage to blood vessels, impacting circulation to the arms and legs, and could require amputation. And then in 5% of cases, that can cause that fatal organ failure or bleeding in the brain. Diagnosis can sometimes be difficult because the symptoms may vary a little bit, but doxycycline is another, again, treatment of choice here, and needs to be done relatively quickly. So hopefully the correct diagnosis can be made early on. And here this just kind of compares both of those two tick-borne diseases. There is a chapter on systemic fungal infections. Or not a chapter, but a section. I did skip those, and we're going to go into the protozoa. Oh my gosh. Protozoal, ZOAL, parasites. I'm having a heck of a time here. I got my thing in the way. Parasites. Okay. Malaria was discovered as in ancient cultures back as far as 2700 BC. So long time ago. And in the late 1800s, during that golden age of microbiology, Plasmodium protists or protozoa was decided that it was the causative agent of malaria, and it's transmitted by Anopheles species of mosquitoes. So a little different than Zika. There are 5 Plasmodium species that can cause malaria, but most of them are going to be Plasmodium falciparum or Plasmodium vivax. Malaria in the US is pretty well eradicated, but up until 1951, it was a problem, and it caused some issues with the Revolutionary War, the Civil War, and things like that. A lot of deaths occurred because of malaria in the southern parts of the United States. But in the 1950s, and maybe even a little earlier, there were measures taken to drain areas that had standing water and certain swamps, and doing mosquito control and educating the public to control mosquitoes. And now pretty much in the US, malaria is brought in from somewhere else. It's an imported case. Most of the cases of malaria occur in Africa, but it can occur in Southeast Asia or the eastern part of the Mediterranean. 2015 saw 212 million cases and 429 deaths. And if you've been following the coronavirus thing, one of the medications that they're promoting to perhaps treat the coronavirus is actually an anti-malarial drug. So there's kind of a tie in there. All right, so that concludes our lecture. You guys excited? Again, just let me know if you have any questions about the final or finishing up the course. Other than that, we will see you guys back for the final sometime on Wednesday. If I don't talk to you personally, I have enjoyed having you guys in class. I wish we could have finished the semester together. I think you guys were an awesome class, and I had a lot of fun things planned. And so unfortunately we didn't get to do those, but good job hanging in there and just persisting through this semester and making it through the end. So I'm proud of all of you guys, and I hope, wish you good luck as you move on to your next adventure.